Okay. Well, let's go ahead and get started for the day. So, several announcements. Remember that quizzes 1 through 3 are due tonight if you haven't already done them. So, quiz 3 will be over the material that we've covered in class. So quiz 1 and 2 are more review type material. So, hopefully you've already done those. Quiz 4 will be due a week from today. Okay, but we haven't covered any of the material on it, so certainly don't go to it. I mean, you can if you want to, but you get 2 opportunities. But we'll cover the material over the next several days for that. Okay. Along those lines, I think that considering where we are right now, my expectation is that probably Wednesday of next week we'll spend the entire day doing a group work. Okay, I want you to bring a computer or a smartphone or something like that. You'll need to be able to access information from the web. And that day will be essentially you come here, I'm going to hand out some things. You're going to pair up in groups of 3, so start to look for that group of 3. And then you'll spend the entire day. I'll be walking around and you'll be essentially working on that worksheet. Okay? So I hope to do that on Wednesday. Okay, that'll be 1 of 5. Those are each worth 10 points. So you'll definitely want to be here. Okay, and then finally, recitation sessions start this week. Zach, the graduate TA, Zach Hazlitt, is going to be going on Thursday tomorrow, starting at 4 o'clock in Walnut 103. The other Zach is going to actually he'll be Fridays 5 to 6, except for this Friday. He's going to start at 4:15. Okay, and he'll be in Walnut 103 as well. So Friday 4:15 to 5 will be short because he's got some responsibilities at 5. And then finally, Ben will normally do it at 5 to 6 on Sundays, but this Sunday he's going to do it at 3 to avoid conflicts with some football event going on. Okay, that's going to be MRB 312. Okay, all right. So our riddle: what occurs once in a minute, twice in a moment, and never in 1,000 years? The letter M, right? Okay, I know. It was easy. It was okay. All right, that's not what we're doing today. Okay, so on Monday we almost finished lecture 1. If you remember, in lecture 1 we are walking our way through metabolic regulation to start off with just its importance. It is very important in regulating steady states, of course. And then we looked at the design of a pathway, specifically the fact that there are these irreversible steps where regulation will occur. And then we spent the rest of the time talking about how can that regulation at those enzymes that catalyze those irreversible steps occur. And we started with a discussion on essentially just substrate availability, where cooperative enzymes are going to be the more important type of enzyme in that versus the Michaelis-Menten enzyme, because they are more sensitive to changes in substrate availability, and therefore their rate will go down or up to much greater degree over smaller changes in substrate concentration as compared to Michaelis-Menten enzyme. Right? After that, we looked at allostery and allosteric proteins. Those can have fairly drastic changes in rate without any change in substrate concentration because they can bind to these allosteric effectors, activators, or inhibitors. And then we talked about covalent modification at the end of Monday and finally left off here, which is our last means of regulation of those enzymes that catalyze irreversible steps. And this is a big one. It's a really big topic, gene expression and regulation of gene expression. As biochemistry majors, most of you are biochemistry majors, there are entire courses in our curriculum that will cover gene expression and the regulation of gene expression. In particular, BC 463 will be the senior treatment of that, or the advanced treatment of that topic. Clearly, I don't want to spend a lot of time on that then, because you'll get more information. And you may have had some other molecular biology and genetic courses. Certainly Life 201 talked about some regulation of gene expression. Okay, but essentially the idea of this is that enzyme concentration will change within the cell over time, whether that be an increase in the concentration of that enzyme or whether that be a decrease in the concentration of that enzyme. That can be done by regulating a number of different steps in what would be considered gene expression. The first one would be probably the most obvious and one of the more well-studied one, and that is the regulation at essentially the transcriptional level. Okay, and what we have down here is just a figure from your book. Oftentimes transcriptional levels of genes will change based off of some extracellular signal that will then propagate through the cell, activating some transcription factor, and that transcription factor will then go into the nucleus, bind to the promoter, recruit the necessary machinery to then carry out gene expression, and therefore mRNA transcripts of that gene will increase. Okay, that's one way to do this. Another way to do this is past that, and that would be to regulate the stability of the mRNA. All right, so once you get to this, mRNA can be broken down fairly rapidly within the cell or not so rapidly within the cell, and there are ways and means in which mRNA can be regulated or can be stabilized, if you will. Okay, so that would be another way to do this, either again to increase or decrease expression of a gene. And then, of course, the next step would be translation, actually making the protein. That would be essentially right here. Okay, that can be regulated, upregulated, downregulated, and of course that's going to lead to the production of the enzyme. Okay, now the final thing would be this. There are, of course, pathways in the cell where a protein is destroyed or degraded. And those can be either slowed down or activated for a particular protein. All of those, at every single step along the way, obviously there is a lot of biochemistry to be talked about and mechanisms for that, but each one of these can either upregulate or downregulate the expression of a particular gene. Okay, and that then gene, especially if it's going up in concentration, that will then increase flux through that particular pathway, or if it's going down, of course, it will decrease flux through that pathway. Right. One of the pathways that we will discuss in this class that illustrates this, I mean, lots of them do, I'll say that, but one specifically that we'll mention is gluconeogenesis, the production of glucose in the liver cell. Okay, so response to this in terms of, if you were to compare this to allostery is very rapid, almost instantaneous. Covalent modifications take typically a little bit longer because there's usually some extracellular signal that propagates a cell signaling pathway that's usually in the minute time scale. This one can be hours, possibly even days when you start to see the full response of it. Okay, so this can be really slow in comparison to the others. Okay. Right. And that's it in terms of lecture 1 in metabolic control. Are there any questions on this slide or any of the other things that we've talked about up to this point? No? Okay, great. Well, let's do this now. Let's shift gears and really, we're stepping into the next unit of class, of the class. In lecture 1, we were really dealing with some very general things. Metabolic control happens in all metabolic pathways that we're going to discuss. So we talked about it very generally. We're now going to shift gears and start to talk about carbohydrate metabolism. Lectures 2 through 6. These topics will take us through essentially almost spring break. So we're going to be talking about carbohydrate metabolism for some time now. There's a lot of important aspects to carbohydrate metabolism that would lay the groundwork for that. It'll pay dividends later on when we talk about other central metabolic pathways. We are going to talk about, in terms of metabolic pathways and carbohydrate metabolism, glycolysis, gluconeogenesis, glycogen synthesis, and breakdown. Those are the 4, if you will, main pathways that we'll discuss. We'll spend more time, though, thinking about regulation. Regulation of carbohydrate metabolism is fairly complex, as you're going to find out. It's complex because it can be very different in different cell types. Not only that, but it also has a bearing on glucose concentrations in the bloodstream. And a mammalian system, humans in particular, right, we have to maintain a really constant supply of glucose in the bloodstream. concentration of concentration of glucose in your blood is maintained at a fairly consistent 5 millimolar. Right? Below 4 millimolar, you get into a hypoglycemic state. Above 8 millimolar, you get into a hyperglycemic state. It doesn't get much beyond that range. In terms of the low concentrations, hypoglycemia, that can lead to coma and even death. Right? The reason being that about 75% of the body's daily usage of glucose is used by the brain alone. So if you start to run out of glucose in your blood, your brain is not going to respond well. Okay, it's going to start to run out of energy, and that's going to make you, well, to put it nicely, tired. Okay, or in a very dangerous situation. Okay, so that's very important. Of course, blood glucose and the regulation of that in our bodies is a very important topic from a health standpoint, simply because of the prevalence of the number of people that are now contracting type 2 diabetes. So especially in Western society. So we'll talk about the mechanisms in that regard as well, because of its important impact on a fairly significant population of the world in their overall health. Of course, there's type 1 diabetes as well. It's just not as prevalent as type 2. Okay? All right, so that's what we're doing. Now, that's through lecture 6, though. Everything that I just said is through lecture 6. We're not going to cover that here, and certainly not all today. What we are going to do today, okay, we're going to start at fairly basic things here. How does glucose get into the body? And you're thinking to yourself, well, I drink, a Coke or something like that. No. I'm talking about absorption at the small intestine level. So we'll talk about that. And then once it gets into the body, into the bloodstream, how does it then get from the bloodstream into certain cells? And what do those certain cell types do to that glucose once it's there? Okay. What it's going to do to it will only take one step in that. Okay. What it's going to do to it ultimately, we'll talk about really all semester long. Okay. But those are the things we're going to talk about. So in particular, we'll talk about tissue-specific absorption. We'll look at the liver and the pancreas are very similar in the way they absorb glucose and interact with glucose. We'll contrast that to something like skeletal muscle, which is fairly different in the way it interacts with and absorbs glucose from the bloodstream. Okay? All right. And there will be some things that we'll talk about in that regard. Now, carbohydrates. Okay? I don't want to spend more than just this slide on carbohydrates. You should have had some sort of carbohydrate chemistry in your past. Okay? And if you haven't, then I'm going to tell you what you need to know for this class in terms of carbohydrate chemistry. All right, so let's start with this. I mentioned glucose a number of times already, so let's draw out in its linear form glucose. Drawing out the linear form because it's easier to draw that out than the cyclic form. Okay? And that's the only reason. Okay? That's essentially D-glucose. D-glucose is what we care about in this class. Okay? You want to draw out L-glucose? Just switch the position of the hydroxyls at every position. Okay? But that's D-glucose, 6-carbon sugar. It will form a 6-membered ring. Okay? Very, very important carbohydrate in terms of metabolism. Okay? All of the carbohydrate that we would store in our body for a rainy day, if you will, is stored as glucose. Okay? Now, let's contrast that to another carbohydrate, another 6-carbon carbohydrate, just to give you some differences here. I will draw out galactose. Galactose is a diastereomer of glucose. So this is D-galactose. Okay. Also an important carbohydrate that you and I will metabolize and that would be found in our diets. Okay. These two and a host of others that I'm not going to draw up here would be called aldo sugars. And the reason being that they have an aldehyde here. Aldo sugars, when they do form the cyclic ring, they'll form 6-membered rings. Okay? So these both will have 6-membered rings. The only difference between them would be the position okay, of this hydroxyl here. Okay. Structurally, they're very, very similar. Okay. And then finally, I'll draw another one here, one more carbohydrate. Oops. Another important one, especially in discussions on health, that kind of thing. Okay, that would be this sugar, which is fructose. D-fructose, also a 6-carbon sugar. This would, though, be considered a keto sugar. And the reason is because it has a ketone rather than an aldehyde. 5-member ring. That's what this will form. Okay. Fructose is the F word, according to nutritionists. Okay, all right. There's some correlations between diets that are high in fructose and obesity, and then the correlations that has with type 2 diabetes. Okay, so again, the F word. It's also found in very high concentrations in every sugary drink that we enjoy consuming. Okay, all right, so anyway. So those are the sugars. Again, D-glucose is what we'll primarily talk about in this class. We will talk a little bit about, after we get through glycolysis, just showing you how are these metabolized in the cell. The metabolism of these are not significantly different than glucose. The first couple of steps are a little bit different. Okay, sometimes they require different enzymes because of the structural differences, or if they use the same enzyme as glucose, that enzyme will have a different specificity for that. Okay, usually lower specificity, meaning a higher Km. Okay, right. So those are the carbohydrates that will encounter. Glucose and fructose will come up again. Galactose not so much. Right. Now, let's think about this. Okay, from here we'll just talk about glucose and say, okay, these other sugars will do the same thing. Okay, let's think about how can glucose enter the body. This is the high-resolution structure of the mucosal cell lining of the small intestine and the brush border. Okay, some of you are like, yeah, I get it. It's not high resolution. That's why you said it that way. I get it. Can we just keep moving on? Can we go on? Can we do that? We'll get there. Okay, so anyway, obviously this is not a very good rendering of that, but it's okay. It'll work for our purposes today. So you're looking at just one cell that would be lining our small intestine and the brush border of this, okay? So what you have here would be the lumen of the small intestine. Okay, obviously this is where our stomach is going to dump whatever we eat. It's going to get into that, and then that's going to be where the majority of absorption occurs. All right, this would be the apical side of that cell, okay, mucosal cells. The basolateral side of that cell will be illustrated here, okay, and this would be, of course, a blood vessel, okay, and we're not really showing here how those two innervate with one another, and certainly this is not a technical drawing of that, okay. Now, below that, what you see here is a table, okay. And in that table, we have concentrations of 3 components, glucose, sodium, and potassium, that are shown above. Okay, and we see here the relative, either the relative concentration or the actual concentration of these things within those particular compartments. Okay, there's also a membrane potential here that we would have to consider. Let me just draw it this way and think about it this way. Okay. This side of the membrane is positive, this side of the membrane is negative, this side is negative, this side is positive. Okay. And we need to discuss how is glucose going to get into these cells. Okay. So let me do this. Let me ask you a question just to get your brains moving in the right direction, okay? I think this is too difficult of a question, but given the last slide, in which direction will glucose move spontaneously? Give me it's where you think it starts, okay? And where it ends, right? Just spontaneous, meaning that it would go in that direction. If nothing else was happening, what is the thermodynamically favorable direction of glucose movement? That's what I'm asking. Okay, thermodynamically, it's favorable to go from here to here or from here to here, okay, or a combination. So I'll give you a minute 30. Okay, I'm going to go ahead and start this. All right, and then I will go back to the slide just in case you don't have it, so you can look at that. Okay, so you got a minute for 30 from this point. Thermodynamic favorability. Where is it? Exergonic movement of glucose. Talk to your neighbor about it. Feel free to do so. Okay, what do you think? Oh, it's not working. Oh, you have a different question. Well, yeah, a different question. That's no good. Okay. How about now? Is that good? We good? Oh, that's really interesting. Okay, good deal. Okay, so go for it. Now you have a minute, and people are responding. That's good. Yay. About 30 seconds left. Okay, about 10 seconds left. So submit an answer. Okay. Submit something. Check this out as a class. All right, let's see the results here. Okay, so out of the cell, into the lumen and the blood vessel, out of the cell, okay, from cellular to blood, outside to inside, from the cell to the blood, here to here, here to here. Awesome. As specific as you can possibly be, right? The strategy there is, if I'm vague, I'll get the answer right. All right? I can hide things in my vagueness. Okay, so here to here is the right answer. You're absolutely right. It will move from here to here with thermodynamic favorability. Good job. Okay, into the blood, out of the cell, starts at apical and flows down gradient toward bloodstream. Okay, inside the lumen. All right, so I wonder if there's well, let's just leave it at that. Okay, so I think outside of the cell is a really good answer. It's going to move in either direction outside of the cell just based off of thermodynamics. So if you come back here, this is just to remind you about the way concentration gradients work. Certainly at this point in time, you've spent quite a bit of time thinking about, I would hope, concentration gradients. If you look at this, based off of this, okay, this relative glucose concentration, things move from high concentration to low concentration. You know that just from your everyday experience in your own home. Okay, so that's essentially being stated here. We typically, in a small intestine cell, or that epithelial cell lining the small intestine, will have typically a higher concentration of glucose in that cell. Volumes are real small in cells. Right? So compared to the lumen of the intestine, even after a meal, although glucose concentration is pretty high in the lumen at that point, still a large volume, so concentration is pretty small relative to what we found in the cell. So it'll move spontaneously, thermodynamically speaking, out of the cell, either towards the blood, which is what we want it to do, or out into the lumen, which is what we don't want it to do. In fact, we want quite the opposite to occur, especially after a meal. You want your intestine to absorb glucose, okay? Not spit it back out. That would be messy in a number of different standpoints, from a number of different standpoints, okay? So when we look at this, if glucose were a big red sphere, okay, which it's not, but if it were, okay, to move that from the lumen of the intestine into these cells, that's going to require some energy, okay? And so we're going to have to actively transport it. Now, once it gets here, it can go out into the bloodstream down a gradient, spontaneous movement into the blood, okay? But this process here, let me highlight that, okay? It's going to be a positive delta G. Requires work. This would be akin to essentially, right, having a cup of water that had food dye dropped in it, green food dye dropped into it, and it's uniform across the water. This is essentially trying to concentrate all that green food dye out of the water. It's going to require some work. You're going to have to do something. We could do it, but it would require some doing. That's essentially what we're saying has to happen here. Okay. Now, let's go ahead and look at this. Sodium. We appreciate sodium, and we appreciate sodium gradients for this reason. If you look at the sodium gradient, the concentration of sodium in the lumen of the intestine is held at fairly constant 145 millimolar concentration. It's held at a really low concentration in the cell, 12 millimolar. Okay? So the movement of sodium, if sodium were blue spheres, I'm going to draw two of them here. They're blue spheres, the movement of sodium into the cell would be a negative delta G. Okay, can do work. Okay. And that's a great thing, that it can do work. Now, I will say this, it's going down its concentration gradient, it's also going down its electrical gradient. This is an electrochemical gradient. This is positive charges moving into a negatively charged environment. That's favorable. Right? So it is a favorable electrochemical gradient that can do work. Now, we can then add these together, right? And if you couple the movement of sodium with glucose, then overall, delta G is negative for the movement of glucose coupled with sodium movement. Okay, and this is where there's these great things in our bodies called proteins, right? And proteins can do amazing things. Okay? And the protein that does this, is a transport protein, is called the sodium glucose symporter. Symporter means it's something that moves two or more things into the cell or in the same direction. Okay, a membrane transport protein, so an integral membrane protein, that will then move, and you'll notice the way I had drawn this, it will move 2 sodiums for every 1 glucose that it moves into the cell. Okay. Now, in a class like 401, we might want to pull apart, well, how does it do that? What's the structure of that pink blob there and that kind of stuff? And that's important, right? Because it's really not a pink blob. Okay, it has a very specific 3-dimensional structure. It has a very specific mechanism that it does this. It's really actually quite fascinating. The sodium will actually, the glucose will bind to the protein first, and then once the sodium binds, that sodium induces a conformational change in the protein that opens it up to the other side. Upon that change in conformation, right, that opens it up to the other side, the protein loses its affinity for glucose, and glucose diffuses into the cell. Okay, and then it releases sodium, and the conformational change resets, and it goes back and it keeps doing that. Really fascinating. And what we're essentially seeing here is that that energy that's stored in the sodium gradient is driving conformational changes of protein. Okay, and that's consistent with the first law of thermodynamics. We're going to essentially have an electrochemical gradient energy in that, and then it converts that to mechanical energy in the change of the conformation of a protein. And it can do work. Yeah. Cool stuff. Really cool stuff. Okay. Questions on that? If you've taken a physiology class, I doubt that I have a feeling that you went through something like this, probably. So this might be a review for some of you. Yeah. No questions? Okay. Now let me do this. Let me go to another slide, okay? This is just cleaner for me, okay? We just looked at how glucose is entering the cell, glucose and sodium are entering the cell, right? Saw that. Now, here's the important thing. If we keep moving sodium into the cell, okay, 2 at a time, there better be something that's taking sodium out of the cell. Because if you don't, then you're going to lose your gradient real fast. Right? So, that's where the basolateral side of this cell comes in. On the basolateral side of the cell, there is a protein called the sodium-potassium ATPase. Some of you may know it as a sodium-potassium pump. Okay? Sodium-potassium pumps are essentially ubiquitous in our bodies and ubiquitous across essentially every organism that we've cataloged so far. Very, very, very important proteins. Okay? Highlighting the importance of sodium-potassium gradients. But this is going to move, the sodium-potassium ATPase, or pump, moves 3 sodiums out, okay, and it moves also potassium into the cell. Okay, so our gradients that we're looking at here are shown down here. Well, you'll notice what I just boxed in. Obviously, if the movement of sodium into the cell is exergonic, the movement of sodium out of the cell is endergonic. Okay? So this requires work. Positive delta G requires work. We could say the same thing about potassium. Okay, if you look down here, the potassium is moving against a gradient, a fairly steep concentration gradient. Moving into the cell, it's going from very low concentration to fairly high concentration. That also, positive delta G, requires work. Okay. Now, in our last slide, I said that movement of glucose requires work, and then the movement of sodium did the work. In this one, the things that are moving, they're going in opposite directions, and they both require work. So where does the work come from? Well, this is where it gets its name, ATP hydrolysis. So negative delta G for ATP hydrolysis. Okay, add all these together, okay, and you get a negative delta G for sodium potassium movement if you have ATP. Okay. Sodium potassium ATPase maintains and establishes the sodium potassium gradient. It's the workhorse of the cell. Sodium potassium ATPases can account for massive uses of ATP in the cell. If you get into like neuronal cell and neuronal physiology, a sodium potassium ATPase will account for like anywhere between 70 to 90% of that cell's use of ATP. A lot of it. Okay. Very active in those environments. Very active here as well. Okay. And we'll highlight this when we get to amino acids, and we can highlight it here for even more carbohydrates. We talked about glucose, but fructose, galactose, other carbohydrates, amino acids, all of them enter into these cells using a very similar setup. They'll have different symporters, but they'll still need the sodium and potassium gradient to do this. Okay? Right. Now let me say this. Let me say one. Just put some labels on all of this. Okay? The sodium potassium ATPase and all of what we see here, this is what we call primary active transport. Oops. We won't talk about the mechanism of how sodium potassium ATPases do this. It's interesting, somewhat complex, but we're not going to talk about it. But that's a primary active transporter, and it's carrying out primary active transport. Okay? If we were to go back one slide, let me go back here. Don't worry, I'll come back to that slide. Okay, but this and the sodium glucose symporter, this is a what we call secondary active transporter or active transport. Active meaning it's moving things against a gradient. In this case, secondary. It's secondary because it's getting its energy from a gradient that was established by a primary source. ATP hydrolysis in this case. Okay. Generally speaking, secondary active transport doesn't occur without some primary active transporter there. Okay. think that that's it for those two cells. Questions about the connection between these things? Yes?
What does the potassium do?
Okay, so the question is, what does the potassium do? In this setup, nothing. Okay, so it'll do other things that the cell needs. But for the movement of glucose or the nutrient into the cell, nothing. Okay, I will show you this, since you brought this up. There is a potassium channel that allows this to essentially leak back to the other side, okay, so that the concentration of potassium can be maintained within the cell. Okay, but the potassium, at least in glucose transport, is not doing anything. Okay, that doesn't mean that it's not doing anything in other aspects of cellular physiology with those cells, but just not. Other questions? Yeah.
So the concentration of glucose on both sides of the epithelial cells are the same, like you have in the chart, as both low. How do you make sure that it goes into the blood, not back?
Okay, so the question is this, right? You have the low and low here, so these are the same. How do you make sure that this doesn't actually go back into the cell from the blood? Is that what you're?
That and
or
from the cell
into the lumen of the intestine. So in terms of going from the cell into the lumen of the intestine, it wouldn't be possible because there's not a passive transporter found on the apical side of the membrane. Okay, the only type of transporter that would be found on the apical side of the membrane is that sodium glucose symporter, and as long as the sodium gradient is steep, it won't move glucose in the other direction. Okay, because every time, going back to that mechanism, every time it opens up to the inside of the cell, that transporter loses its affinity for glucose, meaning it can't bind it on that side. Okay. So that would be the way to prevent it from going back into the lumen. Now, coming from the blood into these cells actually is not prevented. Okay, so that GLUT2 transporter, get rid of this here. This here, we're going to talk about GLUT transporters here in a moment. Right, that's a passive transporter, meaning that it will move glucose in either direction. It just depends on the gradient. Okay, so if there becomes a scenario in which, right, the concentration of glucose in the blood is larger than what you find in the cell, it could actually go into the cell. Right? Now these cells, I will say this, they depend for their energy source, and they need a lot of it because they use a lot of ATP, the sodium potassium ATPase uses a lot of it. They depend primarily on glutamine metabolism, an amino acid. So, and we'll talk about that when we get to amino acids and amino acid metabolism. But that's where the majority of the energy for these cells come from. So, okay. Other questions? Questions? I got a question for you. Okay. Considering this setup, let me do one more question here, at least one more today. Study the last slide, okay, and tell me what you think would happen if the GLUT transporter was inhibited. That GLUT one that I was just talking about, GLUT2. What if that's inhibited? What will be the outcome of that? Blood glucose levels will experience a decline. Glucose import into the epithelial cell would cease after some time. Glucose import into the epithelial cell would not change, or some combination of the above. Okay, talk about that with your neighbor, minute 45, and be ready to address maybe why that's the case. Okay, and your marks, get set, go. Is it going? Activated? Okay, we're good. All right. in about 20 seconds left, so submit an answer. All right. 5 seconds. Submit your answer. Check this out. See what our results are. Okay, let's check it out. Okay, so that's interesting. Almost an equal percentage of you that say A and B and A and C. So, so A and B meaning that blood glucose levels would experience a decline. B meaning that that would affect then how glucose can even get into the epithelial cell, okay, from the lumen. And then there's almost an equal number of you that would say glucose imported into the epithelial cell would not change, okay. Most, I think, looking at the statistics here, most of you recognize that blood glucose will experience a decline. That's good, okay? Kind of true, okay? We haven't talked about this yet, but there's lots of different ways to make sure that blood glucose levels are maintained without having glucose in your diet, okay? So you would expect, though, a decline. I think that that's a reasonable conclusion. The body would then adjust. It would start to break down glycogen, or it would start to produce its own glucose from protein metabolism. Okay, and we'll talk about those things later on. All right, but let's think about this. Glucose import into the epithelial cell, would it cease after some time, or would it not change at all? Who would like to tell me why they thought it would cease after some time? Does anyone want to give me an answer to that? Yeah. Okay. Okay. So if you didn't hear what he said, he's essentially said this, that he thought it was going to be that they would cease after some time because essentially if you keep moving glucose into the cell, the concentration of glucose is going to keep going up in the cell, but you can only move 2 sodiums at a time. So you're going to run out of energy in that gradient. Anyone want to venture a guess or venture a reason as to why they said this would not change? Yeah.
So the transport of sodium out of the epithelia is not sensitive to the concentration of glucose.
Okay.
So you could have an increase of glucose in the epithelia. You could also still have the same gradient of sodium curves. There would still be an overall negative energy change from the sodium from the lumen entering the epithelia.
Okay. All right. So the statement that was made is that essentially we've got they're independent variables essentially. Moving sodium out of the cell is independent of what glucose is doing. Right. The sodium potassium ATPase, all it has to have is ATP to keep moving sodium out. And so then that gradient is maintained, so we'd expect then essentially glucose to continue to move in, okay? All right, so let's go ahead and look at this and think about this. Those are the two competing ideas, at least of people that wanted to share that. So it turns out that it would be affected, okay? So if you were to stop this, so essentially we're putting a big block here, okay? That will eventually, for the reasons that were actually stated here, prevent glucose from coming in. And the reason is because even though you still are establishing the sodium gradient, the energy required to move glucose into the cell gets higher and higher as the gradient gets steeper and steeper, okay? So as glucose concentrations go into the cell, this here goes up and up and up and up until finally the amount of energy it takes to move glucose from here, the lumen, to here would outstrip the amount of energy found in this gradient. Right. ATP can only move so much sodium out. So this gradient is maintained at a fairly constant level, and you really can't get much more energy out of it. Okay. It'd be like essentially if you think about this room, keep stuffing people in this room, keep stuffing people in this room until we're all just kind of like sardines in here. And until we release a person out this door out here, we can't get anyone else in the room. Okay, it's just not possible. That's essentially what we're saying. And that would be an uncomfortable situation, honestly, in this room. It'd be hot. And, you know, I mean, we'd all be really close to each other, though. Okay. All right. Some of you are saying, let's not try that. Let's not do that experiment. I like the space I have right now, even though my desk is way too close to the chair right in front of me. Just wait until test day. It's terrible. Anyway, okay. All right. So we're going to move away from that now. Does anyone have any questions about that? About that question? Questions about the question? Okay, awesome. All right, now let's do this. Once we get glucose into the bloodstream, okay, from the small intestine, I wanted to show you this because essentially what we have here, this would be our small intestine here. Okay. This is a pretty good drawing of it, I think. Okay. And then it goes into the large intestine after that. All right. But if you'll notice something here, okay, all of this blood and all of these vessels will dump into larger and larger vessels, okay, until finally they'll reach this huge vessel called the portal vein. And the portal vein is going to take essentially anything coming out of the small intestine directly to the liver. And then the liver is essentially going to filter whatever is coming from the small intestine, and the blood in the small intestine is going to filter that and then send it on into the rest of the body. Okay? So the liver is going to experience essentially the glut of glucose coming from the small intestine after a meal. It will also experience, of course, the glut of amino acids coming from that. Fats, though, lipids will actually go through the lymphatic system, so they won't go through the liver first. Okay, we'll talk about those things later on. Okay. Now, the other thing to recognize here is this, okay, and this will pay dividends later on, that the venous system, okay, coming from the pancreas will also dump into a larger vein that will also dump into the liver. So anything that's being excreted from the pancreas will first meet the liver. Insulin and glucagon. And those will become very important hormones that we're going to talk about in terms of regulating blood glucose level. All right, so that's anatomically what's going to happen. The liver, okay, will get signals from the pancreas and also the first shot at absorbing anything from the small intestine. Okay. It turns out that the liver is very willing to share glucose with the rest of the body. It does some interesting things to amino acids, though. Okay, and we'll talk about those later on. Saves our life every time we have a protein-rich meal. Okay, all right. All right, so that's, I think, a very good place to stop. Okay, that tells us at least what happened to glucose now that it's in the body. Now we're ready to talk about what's going to happen at a cellular level. Okay, so we'll talk about this on Friday. Have a good Thursday and a good rest of your day today.